The topic we will be exploring in this video has been trending online recently in April of 2025. Countless news articles and social media posts have been made about the so-called de-extinction of the dire wolves. The company behind this project is the first of its kind outside of fiction and is now valued at more than $10 billion. They claim they will be de-extincting several key species like the dodo and the woolly mammoth. That brings us to our question. How is ancient DNA being recovered and sequenced for the Colossal Biosciences De-Extinction project?
People think it's a scary word, de-extinction. They immediately put us with Jurassic Park, and it's not. We're trying to develop these new techniques in order to save animals, in order to bring back animals that, in some cases, were our fault that we don't have them any longer. It wasn't a climate problem, it was a human problem.
We felt that it was very important for us to bring back the dire wolf.
Dire wolf. Dire wolf.
The dire wolf is the first species that we're bringing back at Colossal.
The dire wolf, which most people think is just a mythical creature, was actually a keystone species that was not only endemic to the United States and mostly Americas, but it was actually culturally important to some of the biggest indigenous early tribes in America. So we thought if we could use it as an opportunity where we can also develop technologies that can help wolves, we thought it was the right first project given how important wolves are to ecosystems.
We're able to use these specimens and isolate this ancient DNA out of them.
For the dire wolf, there was a massive study that did many samples across multiple institutions, and Colossal took 2 of those samples and was able to generate whole genome sequences from each. And so this is incredible for any species, let alone species that have been extinct.
The goal of their study was to try and understand the evolutionary lineage of dire wolves and how they relate genetically to other canids like gray wolves, dingoes, red wolves, and coyotes. The research team analyzed 46 dire wolf fossilized remains from across their ancient range in North America to determine if any of them had enough preserved DNA in order to be considered recoverable. Out of those, 5 samples had enough preserved DNA for sequencing. These were from Idaho, Ohio, Tennessee, and Wyoming. The exact demineralization chemical is not named in the study, but standard ancient DNA protocols typically use EDTA, ethylene diamine tetraacetic acid, to break down the mineral structure of bones. This allows trapped DNA to be released. The researchers followed protocols for retrieving ancient DNA, which are very specifically designed to handle short fragmented DNA, damaged chemical ends of DNA, and very low quantities of DNA. Extracted DNA was converted into sequencing libraries using Illumina-compatible techniques. Illumina is a company that makes machines used to read DNA. Illumina sequencing is a method for reading the genetic code, the A's, T's, C's, and G's, in DNA. It works by cutting DNA into tiny pieces, copying and tagging those pieces with fluorescent markers, and taking pictures as each letter of the code is read, one at a time. The result is a super detailed digital version of a genome. These techniques involved for preparing the DNA for the Illumina machines require 3 stages: repair, fill, and amplify. First, the scientists repair the ends of DNA fragments. Then they add adapters to each fragment. These are short synthetic sequences that allow the DNA to be read by sequencing machines. And finally then, the library of DNA is amplified with PCR, polymerase chain reaction, a technique that makes many copies of the ancient DNA fragments. Each direwolf sample was handled by different labs within 3 separate universities, but they all followed similar protocols. Now let's meet our 5 wolves. First, dire AFR. Dire AFR was collected in American Falls Reservoir in Idaho, and the sequencing was done at the University of Oxford. The second wolf, dire GB, was collected in Giganto Bison Bay, Idaho, and the sequencing was done at the University of Adelaide. The third wolf, dire SP, was collected in Sheridan Pit, Ohio, and the sequencing was also done at the University of Adelaide. The fourth wolf, dire NTC, was collected at Natural Trap Cave in Wyoming, and the sequencing was also done at the University of Adelaide. The fifth wolf, dire GWC, was collected at Guy Wilson Cave in Tennessee, and the sequencing was done here in the United States at UC Berkeley.
When scientists studied the DNA of different wolves, they found something surprising. Dire wolves are very different from other canids, like gray wolves and coyotes. They split from them about 5.7 million years ago. Because of this, scientists created a new genus from them, called Innocent. Sadly, this genus ended when the dire wolves went extinct. Why did dire wolves go extinct? Well, there were several reasons. One big reason was the Pleistocene megafaunal extinction. That was a time when many large animals in North America disappeared. Dire wolves used to hunt these big animals. When those animals died out, the wolves lost their main food sources. Their own body also worked against them. People loved dire wolves for their big size and power, but those same traits may have made it harder for them to survive. They were too specialized for hunting large prey. They couldn't adapt easily like coyotes or gray wolves. Their bodies were big and heavy, with short legs, and they needed a lot of energy to move. They were fast sprinters, but once the big, slow animals were born, they couldn't catch the smaller, faster ones. That made it harder for them to survive in a changing world. And finally, there was also a big climate event called the Younger Dryas, which happened about 12,900 to 11,700 years ago. During that time, the temperature suddenly dropped and it changed ecosystems all across North America. This cold period probably made life even harder for dire wolves. It could have affected their hunting areas, their mating behavior, and even their social group. In short, the world around them changed too fast, and they couldn't keep up.